All right. I think, yes, we are live and the people are still coming in. So just 10 seconds more, maybe. Good. So good morning, good afternoon, good evening, depending on where you are. And welcome to the third and last day of BioC 2021. So I hope you enjoyed the poster session. And in this contributed talk session on single-cell genomics, we'll listen to 4 presentations from Kevin Rue-Albrecht, Kevin Blighe, Xiwen Sheng, and David Risso. And before we start, just as usual, a few housekeeping items. So if you have any questions, please put them in the Q&A tab in AirMeet. And please, since we have four speakers, make sure to indicate in the question who the question is for, and not that we have 2 Kevins. So please distinguish between them as well. You can also upvote other questions in the Q&A tab if you would like that. So the way this will work is that we will hear all the 4 talks, and then we will have a joint Q&A session afterwards. And finally, I should say that there will be a recording of this session available in the schedule just shortly after the session. So with that, I will introduce our first speaker, Kevin Rue-Albrecht, who will speak about Velociraptor, which is an R toolkit for single-cell velocity computation. And we have a pre-recorded video for Kevin.
Hello, and thank you for joining this short talk about Velociraptor, a bioconductor package that provides a toolkit for single-cell RNA velocity analysis. I am Kevin Rue-Albrecht, and I am honored to give this presentation, which represents a collaboration initiated with Charlotte Soneson and Aaron Munn, with recent contribution from Michael Stadler. As a brief introduction to RNA velocity, for those who haven't had the opportunity to perform this type of analysis themselves yet, RNA abundance is a powerful indicator of the state of individual cells. Meanwhile, RNA velocity predicts the future state of individual cells on a timescale of hours. It is often represented as an RNA velocity field, which in this example describes fate decisions of major neural lineage in the hippocampus. However, since RNA velocity is computed for each individual cell, it is also possible to visualize individual vectors, projecting the estimated near future of any given cell. Practically, RNA velocity estimates the quantities of spliced and unspliced mRNA by separately counting reads that incorporate the intronic sequence of expressed genes. The method then leverages the model of transcriptional dynamics of transcription, splicing, and degradation, comparing the balance between spliced and unspliced mRNAs to estimate future levels of spliced mRNA in the near future. This example illustrates the textbook case of circadian-associated genes, where the abundance of spliced and unspliced mRNA at various time points throughout the day increase and decrease in a way that cycles back to levels observed at the start of the day. Qualitatively, one can see in the heat map how the levels of unspliced mRNA generally precede the levels of spliced mRNA observed 3 to 6 hours later. One of the most popular methods used to compute RNA velocity is scVelo, which is available as a Python package. This package provides a toolkit to estimate RNA velocity from estimates of gene-wise spliced and unspliced abundances. The package currently offers 3 approaches to estimate RNA velocity: a steady-state deterministic model, a stochastic model, a dynamic model. I have linked the relevant documentation to read at your own leisure, but briefly, the steady-state model works under the assumption that transcriptional phases, like induction and repression, last sufficiently long to reach a steady-state equilibrium, active and inactive. Velocities are then quantified as the deviation of the observed ratio from its steady-state ratio. The stochastic model treats transcription, splicing, and degradation as probabilistic events. The resulting Markov process is approximated by moments equations. And finally, the dynamical model is the most powerful while computationally most expensive, solving the full dynamics of splicing kinetics for each gene. Now, is Velociraptor a better scVelo, and should you turn your back on scVelo? Well, not at all. Simply put, for those of us who are more comfortable working using R and Bioconductor, Velociraptor essentially provides a Bioconductor package acting as a wrapper around the Python package scVelo. In particular, as a Bioconductor package, Velociraptor uses the summarized experiment data structure as input and output for the analysis, allowing users to forget almost entirely about the Python aspect of the analysis, especially conversions between R and Python data structure. That said, Velociraptor is not only a wrapper around the scVelo Python package. Velociraptor also provides additional functions for downstream processing and visualization, including most recently, plotting functions contributed by Michael Stadler. Here, I want to give a shout out to the basilisk package written by Aaron Lunn. The basilisk package installs a self-contained Conda instance that is managed by the Bioconductor installation machinery and manages Conda environments that can be used reliably by Bioconductor packages. This is key to Velociraptor, which uses the basilisk package to run scVelo in a basilisk environment that provides all the dependencies required to run scVelo. In this slide, I illustrate how Velociraptor uses the basilisk run function to run our internal function .run scVelo in the basilisk environment represented by the variable velo.env. The remaining arguments to the basilisk run function are the arguments that are then passed down to the run scVelo function. Obviously, the users of Velociraptor do not need to worry about that part of our internal code, but I thought fellow package developers could be interested in this example of integration between R and Conda. Coming back to the scVelo function implemented in Velociraptor, Bioconductor aficionados will be happy to hear that the function returns a SingleCellExperiment object that contains the results of the velocity calculations. The input to the scVelo Python method is made of two gene-by-cell count matrices, tabulating the UMI count of spliced and unspliced variants of each gene, respectively. Those matrices can be computed from the raw reads using various tools and strategies. In Velociraptor, those matrices should be stored in the assay slot of a single summarized experiment object that is passed as the input. At this point, I want to give a shout out to Luc Zapia for his cell converter package that is used by Velociraptor to convert results from the Python ANDATA format back into the Bioconductor SingleCellExperiment format. For the coming slides, we are using a subset of a dataset known as Hermann spermatogenesis data, available from the scRNA-seq package in a processed format suitable for RNA velocity analysis. In this case, without going into too much detail, the processed object contains matrices named spliced and unspliced in the assay slot, as well as two-dimensionality reduction results, PCA and t-SNE. With the data nicely packaged in a SingleCellExperiment object, running the scVelo methods through Velociraptor is a single function call. It is possible to choose whether this will apply the standard scVelo workflow, including gene selection, normalization, and dimensionality reduction, or use custom information provided by the user. For example, we illustrate here our PCA result computed earlier in R can be used instead of the dimensionality reduction that would otherwise be computed by the scVelo package in Python. As promised, the output is a SingleCellExperiment object, where the various slots are used to store information converted from the Python ANDATA format using the cell converter package. As a small additional note, the output SingleCellExperiment does not contain all the information present in the input SingleCellExperiment. So we take a moment to transfer over the t-SNE coordinates and the cell type metadata to the new object for later use. Having estimated the velocity of each cell, the last step that separates us from visualizing it is to project the velocity vector for each cell onto an existing low-dimensional embedding. This functionality is provided by the function embedVelocity, which takes a dimensionality reduction matrix and the output of the scVelo function, and returns a numeric matrix containing the projected velocity vectors in that embedding. Note that this function is again a wrapper for the velocity embedding tool of the scVelo Python package. To avoid overplotting for large datasets, rather than visualizing the vector associated with each single cell, it is common practice to summarize that information as a grid of regularly spaced vectors, averaging the trend of cells in each neighborhood. This functionality is provided by the grid vectors function. The resulting grid of vectors can then be easily overlaid on top of the dimensionality reduction result using ggplot2, or in this example, the plot t-SNE function of the scatter package. Velociraptor also provides means to visualize gene-wise RNA velocity. The plot velocity function takes the output of the scVelo function and any number of gene identifiers, and displays a choice of 3 plots, including the phase portrait contrasting the abundance of spliced and unspliced mRNA, the RNA velocity of that gene in each cell, indicating whether levels are expected to increase or decrease, and the current abundance of spliced transcript. Separately, we also have the function plot velocity streamline, where streamlines follow the gradient in the velocity field, illustrating path that cell could follow based on observed RNA velocity in the neighborhood. I will conclude this presentation with links to the dataset and scripts that were used to generate those slides. We used the Hermann spermatogenesis dataset available pre-processed by Charlotte Soneson and available from the scRNA-seq package. The script detailing the pre-processing is available from the first GitHub link, and the source code of the Jaringan slides that I am presenting right now is available from the second GitHub link, including code chunks for the processing and visualization steps that I have demonstrated. With that, I will conclude with credits to all the giants on whose shoulders this work stands, including earlier work in the fields of RNA velocity, but also the entire Bioconductor community for personally motivating my everyday work for just about 10 years now. Thank you.
Thanks a lot, Kevin. So if you have any questions for Kevin, please add them in the Q&A tab. So with that, we are immediately moving forward to the next speaker, who is Kevin Blighe, who will talk about scDataviz, a package for single-cell data visualization and downstream analysis. Please, Kevin.
Yeah, I'm loading the file now. So hopefully that has come up.
Yep.
Yep, great. My name is Kevin Blighe. I'm currently a Chief Science Officer in London. I'm also a visiting professor at Ulster University in Northern Ireland. You see in the top left, I've got the BioStars logo. I contribute a lot back to the community via my role as moderator in BioStars. On the bottom right, I've got the Bioconductor logo. I'm also a moderator in the Bioconductor support forum. And as the other Kevin's just said at the end of his presentation, it's a real great honor, as it always has been, to contribute to Bioconductor in any shape or form. And so my presentation today is about scDataviz. scDataviz is my fourth package into Bioconductor, and I was invited to give this talk by an organizing member. So without further ado, I'll move on. So to understand scDataviz, it's important to go over the history. It's actually my most sentimental package, purely because of its history. So the initial development actually began back in 2015 when I was a senior bioinformatician at Queen Mary University of London. I then moved over to Boston, where I was working at Brigham and Women's Hospital and Harvard Medical School, where some functions that I developed then would eventually go into the final package on Bioconductor. I then moved back to Europe, where I was based at UCL Cancer Institute, and further functions were developed there. And then later at King's College London, if you can believe me, further functions were developed for the package there. And then finally, on the fifth point, so clinical bioinformatics research is my own consultancy in London. That's where I kind of packaged it all up into a Bioconductor package and submitted it. And thankfully, it was accepted into Bioconductor. It's important to remember, though, that this package was primarily developed while I was analyzing and testing mass and flow cytometry data. It does cover RNA sequencing data too, so it kind of caters for both worlds. It sits in a kind of an unusual place, to be honest. So a bit of trivia to lighten up the mood on Friday. After this package was accepted into Bioconductor, I also submitted it to Sean Davis's awesome single-cell GitHub repository. I'm sharing this tweet by Sean with his permission. Basically, I learned that I was actually the 100th independent contributor to Awesome Single Cell. So this is a useful random piece of pub trivia, should it ever come up later down the line in a pub quiz. I doubt it will, though. Anyway. And so, yes, so everything with SC as a prefix is for single-cell RNA sequencing. That's what we're trained to think, right? But it is important to remember that this package was primarily developed with cytometry data. So when you see the SC prefix, you're thinking it must be for single-cell RNA sequencing. And it does cater for that, like I said. Basically, scDataviz, it's built using the SingleCellExperiment class. That's the fundamental packaging unit for the data, which ensures standardization across other functions in Bioconductor and or generally. So technically, yes, it supports single-cell RNA-seq via certain import functions. I would say, though, as it's coming from a cytometry route, which is still technically single-cell, it requires a bit of extra fine-tuning and further development in order to prove its efficiency with single-cell RNA-seq data. I put it to a real test recently, and I was equally heartbroken as I was pleased because I had a 200,000 cell 10x data set. I processed it in SURAT and then I exported it to scDataviz, and it actually catered quite well for this very large RNA-seq data set. It required quite a bit of memory, though, and there's a bit of fine-tuning that needs to be done, basically. I also wanted to keep scDataviz somewhat leaning more towards cytometry, as I realized at the time when I was submitting it to Bioconductor that Dan Boonis was concurrently developing DITO-seq, which caters definitively for RNA sequencing. So I didn't want to step on anybody's toes. I wanted to keep them kind of separate, one for cytometry and one for RNA sequencing. And then so to give you an idea of what the package can do, I'll just give a brief overview of the key functions. Basically, one of the main import functions is process FCS, which just allows you to import FCS files. This also permits downsampling of cells, and the user can apply different transformations to the data. In cytometry, that's usually the hyperbolic arc sign transformation with different factors. So 5 for flow is a standard one, and then 150 for cyto for mass cytometry. And this imports the data basically straight into a SingleCellExperiment object. The contour plot function, this allows you to view regions of high cellular density. And this can be useful to identify, I guess, clusters that could be outliers. So if you look at the plot on the right, for example, that's a large data set of over 400,000 cells. And the cluster density is roughly even across the different islands. Whereas the plot on the left, 260,000 cells, you can see that there's one cluster of extremely high density of cells and the other part of it is kind of less dense. So that's kind of a skewed data set. So you can kind of get that feeling based on using this function. Marker expression, it's kind of obvious what this is doing. It just allows you to view the expression profile of a given gene on a given reduced dimension. So the best example to show here is CD25 on the bottom left. You can see it's highly expressed in the 2 clusters on the top left of the plot, whereas it's not expressed at all in the other clusters on the bottom. And as mentioned, this can be applied to any reduced dimension that's in the SingleCellExperiment object, be that a PCA, TISNE, or UMAP layout. And then so the cluskNN plot clusters function, these are literally using the SURAT's find neighbors and find clusters functions. So they work quite rapidly on even large data sets. And they identify the different clusters quite well, as you see from the plot, as you would expect by visualizing the layout. And so it allows the standard algorithms that are implemented in those SURAT functions for community detection, such as Louvain with multi-level refinement, and then SLM, smart local moving. Marker enrichment function, I think this is what gives the package a bit of an advantage over others, or maybe not, I don't know. It allows you to identify positive and negative genes per cluster. And it outputs also sample metadata cluster abundances. So basically, this can be useful for cell type identification. If you don't want to use one of the automated methods, which can often disagree with each other quite a lot, this function allows you to easily identify which markers are highly expressed or not in each cluster. So, I mean, you can see in the data that I'm showing there are, for example, a few CD3 positive clusters, which are naive T cells, if I'm not mistaken. And then so I've implemented 2 algorithms for identification of these, basically a simple Z-score method and then a quantile method. The quantile method works quite well. Basically, if you set the probability at 0.25, it means that the positive markers will be in the upper quartile and the negative markers will be those that fall into the lower quartile. And so this is the other part of that table. I couldn't fit it all in one slide. Basically, this is the metacluster abundancy. So you see it outputs the percentage of cells per sample per cluster, and that's useful information. For example, you don't want a situation where one cluster is just dominated by 90% or 100% of an individual sample. you may better want an even spread of the cells across all clusters. And then on the right, you can see I also output the number of cells per cluster per metadata. And the metadata here is just the conditions, so healthy and disease. And again, in this case, you may actually want a cluster that has, say, 100% of, say, the diseased cells, and then that would be useful information to have. And then plot signatures. Similar to the previous one, this just allows you to visualize which markers are high or low in each cluster. You can apply your own summarization metric. When developing this, the default is just the mean expression of a gene or marker per cluster. And then the expression range is squeezed to be between -1 and +1, just for visualization purposes. So this is processing the data a bit differently from the previous function, but it actually produces the same results as the marker enrichment function. And that's actually it. So this is really just a short talk. So concluding remarks, thank you to everybody. Thank you to Bioconductor and all the people in attendance watching this. I've listed some key people on the left. So Miles Lewis and Michael Barnes at Queen Mary University in London. Jessica Lasky-Sue is my supervisor in Boston at Channing Division. Javier Herrero was my supervisor at UCL Cancer Institute, and then Shahram and Jessica at King's College London. And so if you are using this, I mean, please contact me with any issues that you find. GitHub pull requests are very welcome. And yeah, thank you very much.
Thanks so much, Kevin. And again, if you have any questions for Kevin, please add them to the Q&A tab. So we're going to move on to the next speaker, which is Xiwen Sheng, who will talk about scRA for large-scale single-cell RNA-seq data manipulation with GDS file.
Okay. Technology develops and decreasing cost of single-cell RNA-seq are leading to a larger and larger number of cell assays per experiment. The scalability leveraging on disk database for our memory processing remain an important issue to address. Here I introduce a new Bioconductor package, scRA, and it provides large-scale single-cell RNA-seq data manipulation using genomic data structure, GDS file. GDS is an alternative to HDF5 and TOOLKIT DBs, and it supports hierarchical structure to store multiple array-oriented data set. It is suited for large-scale data sets, especially for the data which are much larger than the available memory. Data can be compressed and decompressed internally. GDS has been widely used in dual-SNP data, whole genome sequencing data. The R package in the GDS ecosystem includes GDS-FNT, SNP relay, scRA, DOWNSTREAM, and Genesis. In the scRA package, we apply GDS to single-cell data manipulation and analysis. It supports sparse matrix and store the whole single-cell experiment objects into a compressed file. To reduce the existing analysis packages, scRA provides internal C-awards function for delayed arrays and single-cell experiment objects. In this presentation, I will use a small and a large data set to show the function in the package, including PCA and UMAP analysis. Here is the workflow for data analysis. We use a scRA to open the GDS file for single-cell data. The function returns a single-cell experiment object with delayed matrix. Delayed matrix wraps a matrix stored in the GDS file and allow common operation on it without loading the whole object into memory. The downstream data analysis can be done by, for example, scalar packages, which support the single-cell experiment object. The sparse matrix in GDS, for example, count matrix, store and compress non-zero number in column major order. So data reading across gene in the direction of column is efficient. But reading data moving over gene is not efficient. So I rewrite the low specific function in delays matrix steps for faster implementation. For example, low bets, which is used to calculate the variance for each row. The kit function in scRNA packages are listed in the table. The first function can say a single cell experiment object to a GDS file. The second function returns an instance of single cell experiment objects from the file using delays matrix. The third function can use to get a delays array objects from a specific GDS node. Here example. The single-cell dataset for mouse retinas is from the scIMAC package. SC converts GDS is used to create a GDS file. Here is the data structure stored in the GDS file. Feature ID is used for gene ID, sample ID according to cell ID. The variable count is a sparse matrix. Two variables, cell ID and cluster, are associated with sample ID. scExperiment return an object of single-cell experiment, and the object contains count data and current data, cell ID, and cluster. Let me show you an example using a small data set. There are 24,000 genes, 49,000 cells, and 3% non-zero number in the count matrix. The in-memory sparse matrix of the count data takes 450 megabytes. Data size in the compressed GDS is reduced to 40 megabytes. Here example in the books orchestrating single-cell analysis with Bioconductor. A typical workflow for single-cell analysis includes loading data, quality control, dimension reduction for data visualization, and clustering. After loading the data from a file, we have a single-cell experiment object, SCE. In QC, we identify a subset of cells with good quality and compute log transform normal expression on the subset. The next step is to run PCA and UMAP using the log count data. When you have the GDS file, the difference is just the data loading. The count and log count data in the new SCE variable from GDS are the sparse form of delays matrix. Here is example is the next example is a large data set with 1.3 million mouse brain cells. There are 7% non-zero number in the count matrix. The in-memory sparse matrix of the count data is predicted to use 30 gigabytes, but it cannot be loaded into memory using the sparse matrix defined in the matrix package, because the number of non-zero is out of the limit. The file size of compressed GDS is about 3 gigabytes. That is 10 times smaller. As similar as the small size example, we open the GDS file, compute log transform normalized count data, and then run PCA and UMAPs using the log count data. In the step of normalization, log count data is a delays matrix. Well, it is not stored in memories or a file. The number in the log count matrix are calculated only when they are needed in the next step of calculation. In PCA, randomized algorithm is used to reduce the computational burden without losing the precision for top principal component. By default, 500 genes with largest variant are used in the scatters function run PCA and run UMAP. I will show you the running time and memory usage with different number of genes in the next slide. The benchmarks were conducted using the large data set with 1.3 million cells. The RX hours and memory usage are shown in the figure with different number of genes used in PCA and UMAP. In general, the running time and memory use both increase linearly with the number of genes in the calculation, except the CPU hour for UMAP. Overall, the running time are less than 4 hours, while the memory usage is, however, is even larger than 100 gigabytes for 2,500 genes. I use the run PCA and run UMAP function in the scatter package. So the running time and memory usage so here only reflects the implementation in the scatter package. In summary, the scRNA package is still in early state of development. GDS is used as a file-based representation for single cell count data and delays array backend. It is suited for large-scale data storage and manipulation. scRNA is designed to leverage existing R packages by providing single-cell experiment and delays matrix objects. The downstream package, for example, scatters, can run analysis on GDS file. In our future plan, we will further integrate GDS in scRNA with Bioconductor infrastructure, especially with the GDS arrays and delays matrix stats package. We will also consider re-implementing some memory intensity algorithm for even larger data set. I would like to thank my colleagues at the Genomics Research Center at Blighe, Jean-Liu from Roswell Park Comprehensive Cancer Center, the cluster in the National Center for Supercomputing Application and CSA at UIUC was used in the benchmarking. Thanks.
Thank you very much for that. And again, if you have any questions, just put them in the Q&A tab. So before the questions, we will listen to our final speaker, which is Davide Risso, who will tell us about the graphical model for single-cell RNA-seq data. And here, again, we have a recorded video.
Hello, everybody. My name is Davide Risso. I'm from the University of Padua, and today I'm going to talk about a graphical model for single-cell RNA-seq data. So the problem is that single-cell RNA-seq is quite complex data. So we have that neighboring cells influence each other, and more importantly, even genes within a single cell show complex correlation patterns and dependency structures. And what is also clear from these pictures is that graphs and networks could be a good way to represent and study those dependency patterns. So the system that we study, and that was sort of the motivation from this work, is the mouse olfactory epithelium. This is a fairly simple system in which we have only a few cell types, the most important of which are the horizontal basal cells, which are the stem cells of this system, and of course the olfactory sensory neuron. So we started to study this system back in 2017 when I was collaborating with the John Nye lab at Berkeley. So we only had a few hundred cells, but we were still able to sort of detect this sort of, or discover this sort of pattern of development from the olfactory stem cells, the HBCs, all the way to the mature cell types of the system. And in particular, we are particularly interested, of course, in olfactory sensory neuron and neurogenesis. So now, a few years later, we have essentially redone the same experiment with the 10x technology. So we have many more cells. We have 25,000 cells for this experiment. And this is a UMAP representation of those data, where we can see that we have a good numbers of cells, and we were able to identify all the major cell populations of this system. So the HBCs, or the stem cells, that gets activated, they turn into global basal cell, which are sort of an intermediate stage before neurogenesis, and then we have immature neurons all the way to mature olfactory sensory neurons. So one of the goals of this experiment is to look for dynamic expression of genes across development and across these trajectories. So what we did back in the first paper was to sort of look at transcription factors that may be responsible for these type of patterns. And we created a network of transcription factors using simple correlation analysis, where we identify essentially 3 groups of transcription factors that here are color-coded by the cell type in which they are mostly expressed. And so you can see all of these transcription factors are more active in stem cells, and these ones here are more active in neurons. It's quite clear that a network is a good structure to study this data. We're thinking about maybe formalizing this in a graphical model, in a graph. So let me give a more formal definition of a probabilistic graphical model. This is, I promise, the most boring slide of my presentation. But essentially, a probabilistic graphical model can be defined as the pair GF, where G is our graph with V nodes and E edges, and F represents a family of distributions. So if we want to use like a graphical model, we have to choose 2 things. First of all, we have to choose a family of distributions for our data, and then we have to have a graph, or if we don't have a graph, we have to estimate it from the data. In our application, we decide to specify a model that is the more general, as general as possible. So we specify a model that has a node conditional distribution of a zero inflated negative binomial with certain parameters that you can read here. And once we have these sort of node conditional distribution, the problem of estimating the graph, or the description of the graph, is essentially a regression problem, in which we can say that in this case both the mean and the zero inflation parameter of our distribution for gene S depends on all the other genes in a certain neighborhood of S in the graph. So now that we have the distribution of the random variables, all we need to have is the graph. And so the problem is, in this context, we don't really have a given graph, so we have to estimate the graph from the data. And this is called structural learning in this type of literature. And one approach is to start with a complete graph, the graph that has all the edges and all the connections among nodes, and take sort of a statistical hypothesis testing approach, in which we define the null hypothesis that all the regression parameters of the 2 regressions, that of S on T and a T on S, are equal to 0 for all the possible subsets of our set of vertices minus the one that we're testing. And so if we do not reject the null hypothesis, then we have a good confidence that the edge can be removed from the graph. And we can proceed like that in a local algorithm that is known as the PC algorithm. So does that work? Well, to test it, we did some simulations, and in this case, we were interested in, first of all, in seeing if the approach and the algorithm works. So we have the F1 score of the, comparing the true edges and the estimated edges, and we were also interested in the node conditional distribution. So we simulate data from the Poisson, the negative binomial, and the zero-inflated negative binomial. And we consider sort of 4 different algorithms that specify the model as a Poisson, as a negative binomial, and as a zero-inflated negative binomial. You can see that, of course, for like Poisson data, all the models work well. For negative binomials data, when we start having over-dispersion in the simulated data, the Poisson doesn't work as well anymore. And when we start having zero inflation in the simulated data, of course, the zero-inflated negative binomial works better. So I'm saying, of course, but actually this is true only when we simulate counts with a large mean, so we have large counts. When we simulate data with small counts, though, we see that things are quite different. And in particular, now all of our algorithms' performances are essentially the same. So it seems that even if we introduce over-dispersion in the data, and even if we introduce zero inflation in the data, even a simple Poisson model works as well, as long as we have small counts. So I think this is a very interesting result, because I think it can contribute something to the ongoing debate on whether single-cell RNA-seq data are truly zero-inflated or not. So I think a lot of people have seen that for UMI-based technologies, we don't actually need to model zero inflation in the data, and I think this has to do with the small mean situation that we have here. With UMI platforms, you have smaller counts. With smaller counts, the distribution gets sort of brought back towards zero, and it seems that zero inflation is not a problem anymore. So let's go back to our data and to our problem and see if we can actually help understanding transcription factor dynamics across development. To do that, essentially, we apply our approach to different stages. So we start with the activated HPCs, which, as you may remember, they're the activated stem cells of this system. And we have essentially these graphs. The black points are hub nodes in this graph, so nodes with a high number of connections. And the graph was we detected some communities in this graph with a community detection algorithm. So essentially we clustered the nodes in the graph. And then for each of the clusters or each of the community that we found, we ran a gene set enrichment analysis to sort of interpret the meaning of that community. And in this case, you see that we have cell cycle, epigenetics, and epithelial cell differentiation as important terms in the gene ontology. And then as we move on towards neurogenesis in the GBC stage, we see cell proliferation, signaling, expression, regulation, stem cell differentiation, and p53 and DNA replication start to play a role. And then when we move to immature neurons, we see that neurogenesis now is the biggest term. And then as we reach the mature neuron stage, we now have chromatin and TGBF beta signaling. I think I finished my time, so I just want to acknowledge my co-authors in this project. Kim, who is a postdoc in my group, Koen, who is a postdoc at University of Ghent and at UC Berkeley, and Monica, who is a professor at the University of Bologna, and also CZI and NCI for funding. And thank you for your attention. And if you're curious about our results and you want to read more, we have an archive paper with all the results. And these methods are implemented for now in a GitHub package called Learn to Count that you can find at that address. And I plan to eventually include these functions into the ZimBee-Wave package in Bioconductor. So stay tuned for updates.
Thanks so much, Davide. And again, if people have questions that are not about how you created your video, please put them in the Q&A and we will get to that. So let's start with the questions, and I will start reading here from the most popular ones. So we have a question for Kevin Rue. So very useful, thanks for Velociraptor. Are there any plans to do something similar with cell rank, which is for trajectory inference in single cell data?
So yeah, when Velociraptor started, I think we started with the ambitious goal of having it as a wrapper to many methods, and over time we kind of focused already on one to get, let's say, the proof of concept running. But yeah, we were open still to that goal, and I'm actually currently helping on a project that involves RNA velocity. So even more motivation for me to revisit a bit other methods. But yeah, I even tested using the scVelo method on a data set, and I'm quite keen to essentially try a few other things.
Perfect. Thank you. So let's go to the next one. I will take this one for Kevin B. So does scDataviz work with out-of-memory data like HDF5 and similar?
Not directly, no. But if you can get your HDF5 object into a SingleCellExperiment object, then that works. So I'm pretty sure there's a package on CRAN or Bioconductor that can do that somewhere. But yeah, I mean, that would be a useful development to add to the package in the future. Thanks.
Great. So there are two questions for Shuwen, which are fairly similar. I will show this one. So there are many different disk backends in or available to Bioconductor users, GDS, HD5, TileDB. Can you comment about differences in performance? And there was a similar question, which was whether the package was benchmarked against HDF5 and TileDB, for example, for speed of PCA and UMAP.
Yeah, that's a very good question. So we did some comparison, but what I can say is the first comparison between GDS, HDF5 and TileDB, well, it's not easy. So for example, I try HDF5 for PCA and UMass. I found it's very, very slow. But I mean, when after checking the code, I mean, the issue is not related to the data format itself. I mean, it's just because the developers do not provide efficient implementation. For example, the implementation for low variant calculation. So I mean, at least for the example PCA and UMass, what I can say is that they should be very similar, the performance. If the developer can provide efficient implementation for each backend.
Okay. Thank you. I will mark this too. So I had a question for Davide. Do you have any kind of limits on the number of cells that you need for your graphical model to work well? The expression level, I mean, your transcription factors may be lowly expressed, and that's also fine. Or do you have to do some pre-processing of the data specifically to make this work nicely?
Yeah, yeah. So the number of cells is not much of an issue. Well, I guess it is, but as long as you have, I guess, more cells than well, I guess then genes that have connections, you're fine. But of course, as you increase n, your power goes up, and the power of the tests go up, and so everything works well. Basically, we're doing something sort of the opposite that what people usually do. We're not rejecting the null hypothesis, but we're sort of failing to reject it. So if you have low power, it won't work, right? So you kind of need a lot of cells, and that's why sort of we waited to have like a 10x genomics data with a lot of cells, because otherwise it's not. And then in terms of pre-processing, we usually do like, well, actually we don't do any pre-processing at all. We do like the usual kind of normalization for sequencing that you can put as an offset in the model as usual, and that's it. That was actually the main motivation for this work, to have a model that work on counts. Because of course there are many like graphical models on like Gaussian distributed data, but that requires pre-processing.
Yeah, thank you. So we're almost running to time, but just a quick question here for Kevin B. So other than the input data format, how and why does cytometry data differ from the regular single cell, which I guess is single cell RNA-seq in terms of normalization and analysis?
Yeah, I think that's from Jenny. That's a difficult question. So all I can say is with cytometry, CyTOF data, for example. So CyTOF data, I guess one should first of all expect zeros to be quite frequent in all types of data. Although I'm cautious to say that RNA-seq is zero inflated based on David's talk. But so CyTOF data, one should expect zero inflation and also negative values as well in CyTOF. And the transformation that we apply to that is essentially like a log transformation, but it can deal with the zero values and the negative values at the same time as it deals with the positive values. Whereas that in single cell RNA sequencing, we apply different normalization methods. It's a genuine difficult question to answer, though. I've not explored it extensively. If anybody else can chime in, that would be appreciated. But yeah, that's kind of stumped me, that question.
That's all right. Okay, just a very quick question for Kevin. Wouldn't it be preferable to keep all the components of the input single cell experiment in the output from scVelo?
Yeah, so the reason why the output doesn't contain everything from the input is because of this conversion from R to Python. So since we give the Python side only what it needs to run the method, the matrices and things, so Python then generates its own object, and the output is the direct conversion from the Python object. So if we try to put back what was in the input object back into the output object, we would be taking a risk of overwriting stuff that the Python side has sent back. So it is feasible, but it's a little bit on the risky side. So I'm leaning on the side of saying, okay, here's the output that you requested. If you want to transfer stuff over, we'll leave it up to you.
Yeah. Great. So I think we actually made it through all the questions, and we're just on time. So thanks again to all the speakers. This was were great talks. Thanks to the audience for engaging in the discussion. And I'll see you in the next session. Thank you.